chromosomes are in the nucleus of each cell. The DNA molecule is packed into the to thread-like structure is also called as the chromosome. Chromo means color, different types of the color. Main, the nucleus of each cell, the DNA molecule is packed into thread-like structure is also called as chromosome. Chromosomes, the first described by the Strasburger in 1875. The term chromosome was coined by Waldeyer. Okay, the term chromosomes was coined by Waldeyer in 1888. Each chromosome is made of the DNA, tightly coiled proteins. The coiled protein is also called as histones. What are the importance of the histone? These are the supports. It's a structure. Okay, histone proteins also they support a structure. Chromosomes are not uh, visible in the cell nucleus, not even under the microscope. These are the cell not dividing. DNA that makes a chromosome becomes the more tightly packed during the cell division, and it's in the visible under the microscope. Actually, these are the seen under the microscope. There is no seen to the directly visible. Okay, these are all seen to the under the microscope, especially the can use for the electron microscope, different electron microscope. Okay, women cells have 46 chromosomes, 23 identical pairs, 24 actually 22 pairs of the autosomes, one pair of the sex chromosomes. Autosomes are the chromosomes which are not involved in the sex determination. Actually, allosomes are they can involve in the sex determination. Autosomes they cannot participate in the sex determination. Only allosomes are the participate in sex determination. For example, different organisms, number of chromosomes also, human beings, especially 46 chromosomes. Next one is the roundworms are the Drosophila melanogaster roundworm. Also, how many two chromosomes, and especially in plant cells, different types of the plants. Onion, they are containing the number of chromosomes in 16. Fungal mold 16, carrot 20, tomato 24, and next one is the tobacco 48, rice 24, maize 22. Another important is the haplopappus. The lowest chromosome also seen to the haplopappus. Haplopappus in the two chromosomes. These are the very important. These are the structure of the chromosomes, double helix of the chromosomes. Especially see these are the diagram is related to the chromosomes. P arm and Q arm, both are especially they are the constructed and the link between the centromere. Okay, double helix DNA double helix. These are the chromosome differences of the chromosomes. These are the two arms, especially two chromatids, especially these are the short arm is also called as a short arm. Next one is the long arm. Okay, these are the two chromatids. Okay, in between the attached by the centromere. What are the structure of the chromosomes? Centromere, it is a part of the chromosomes that link sister chromatids. Especially these are the part of the chromosome is linked with the sister chromatids. Centromere gives the characteristic shape to the chromosomes and helps to describe the location of the specific gene. Let us discuss the P arm. P arm is also called a short arm. And these are the P arm and Q arm. Okay. P arm is the short arm which is the present in the upward portion. Is the present in the upward portion. Q arm is a long arm which is the present in the downward portion. Okay. The both are they can connect with the in centromere. P arm, Q arm. P arm is also called as short arm. Q arm is also long arm. Okay. Next one, telomere. What is the meaning of the telomere? It is the present in the tips of the chromosomes. What are really telomere end part. Okay, it is the present in the tips of the chromosomes. Next one is the chromatids. What are the chromatids? Okay, what are the chromatids? Telomere chromatid is two identical copies formed by the replication of two chromosomes. What is the meaning of the chromatid? Chromatid means two identical copies formed by the replication of two chromosomes is also called as chromatids. Chromatins. What are the importance of the chromatins? Chromatin is a complex of DNA and protein found in eukaryotic nucleus, which is packaged chromosomes. The structure of the chromatin varies significantly between different stages of the cell cycle according to the requirement of the DNA. Chromatin is a complex of DNA and protein found in the eukaryote nucleus. Chromatin is mainly the eukaryote nucleus. Okay, so what are the types of the chromosome? There are the four types of the chromosome: telocentric, acrocentric, submetacentric, metacentric. I will discuss the each and everything. Okay, the first one is the telocentric. What is the meaning of the telocentric? Telocentric mean telocentric is centromere is present at the end of the chromosome. See this diagram also. The two arms is there. One is the long arm and another one is the short arm. Especially mainly centromere. Centromere. The telocentric is mainly centromere is present at the end of the chromosome. This is also called as telocentric. Telo mean end. Telo mean end. Next one, acrocentric. What is the meaning of the acrocentric? The centromere is the almost terminal portion, end portion. Especially the telomere is the end portion. Is also called as telocentric. Acrocentric means especially the centromere is almost terminal portion. It is one large and the another very is the smaller. Uh, Submetacentric. These are the acrocentric is the eye shape. Submetacentric, the centromere is not at the middle position of the chromosomes. The arms, the arms are the unequal position. The shape is the L-shaped appearance. See, submetacentric. Next one, metacentric is the V shape. Is that the centromere is at the middle position. The arms are the equal position also taken into the metacentric. Okay, metacentric is the equal portion of the arms. Submetacentric is the unequal portion of the arms. These are the metacentric, submetacentric. Next one. One minute. Okay, next was the chromosomes. 1 minute. Okay, types of the chromosomes, autosomes, allosomes. Already we discussed. Autosomes are the 22 pairs of the autosome. Autosomes contain the non-allosomal chromosomes or the genetic heredity information. It is the homologous chromosomes. They are not. They are the morphologically similar. In women being the how many autosomes? 24, 22 pairs of the autosomes are 44 of autosomes. Autosomal abnormal. What are the mainly is there some abnormalities of the autosomes? Abnormalities. The most well known is the most common autosomal abnormality is the Down syndrome. Down syndrome is one type of the syndrome. These are the some of disease. Down syndrome in many the people is the Down syndrome have the irregularity with autosome pair 21. Autosome pair 21. The most cases is the extra chromosome, a trisomy 3x, trisomy condition 2n. 21 is the is the more rarely 3 to 5% there is the structural and modification in these chromosomes. Especially there is the translocation of all are the part of the chromosome 21 chromosomes, the 14 for Down syndrome, especially what main 21 chromosomes is the extra chromosomes. The actual genes of the chromosome 21 that are the responsible for the Down syndrome are now being the identified. That these are the at least that 350 genes involved. About the 2 to 4% of the people with Down syndrome are genetically mosaic. Genetically mosaic. That is the sum of the their cells have the chromosome 21 trisome, while others do not, resulting in the generally milder systems, milder symptoms. The translocational type of the Down syndrome is also as the less service of the symptoms. Next one, Down syndrome also the first described by the John London Down physicians. These are the physician, and these are the include the epilepsy, hypothyroidism, cross eye eyes, near sightness to far sightness of the cataracts, hearing of the empowerment, and that is the makes the difficult process of the auditory information, heart defect, intestinal mal interest, inter, intestinal malformations, herniasis, and the marked successfulity in respiratory infections such as the pneumonia. These are the all are the Down syndrome disease symptoms. Trisome, especially trisome, is the 13th trisome. Okay, these are the Down syndrome. Next one, mainly the autosomes. Next one, allosomes. Already we discussed the allosome. One pair of the allosomes are present in the human being. It is the heterologous XX and the XY, male chromosome XY, female chromosomes XX. Okay. Next one. What are the female sex chromosomes abnormalities? Turnover syndrome. Down syndrome is the autosomal abnormality. Autosomal abnormality. The Turner syndrome occurs the females inherit only one X chromosome, their genotype. If they survive the birth, these girls have the abnormal growth of the parents. Okay, abnormal growth of the parents. They they are they are short in the stature of the average of 4 foot to 7 inches in the adults, and often have the distinctive webbed necks, the small jaws, and the high arched plates. These are the characteristics. These are the characteristics of the Turnover syndromes. And these are generally lack prominent female secondary sexual characteristics. Okay. Women with the Turnover syndrome is the higher than the average incidency of thyroid disease, vision and the hearing problems, heart defects, diabetes, and the other autoimmune disorders. In few individuals, there is a slight mental retardation. The Turnover syndrome is the in rare. Okay. Next one is the current estimates of the frequency range from 1 to 20 or 1 to 50,000 female infants if diagnosed in the early childhood. Regularly injections, the human growth in the hormones can increase the stature by the new inches. And the beginning around the normal age of the puberty, estrogen replacement therapy can result in some breast development of the menstruation. This is the treatment allow the Turnover syndrome women appear the relatively normally. Okay, next one is the triplex syndrome. It occurs women the inheritance X chromosome. There is the genotype triplex syndromes. Mainly are the as adults is the super females or meta females as they are the sometimes known as the generally are the inches are so the taller than the average with the unusual legs and the slenders to shows the otherwise the appear normal. They usually have normal development of the sexual characters and the fetal, but the tend have some ovary abnormalities that is the lead to the premature ovarian failure. They may have the slightly learning difficulties, especially in speech of the language skills, and usually in the low range of the normal intelligence. The triple X syndrome is the less rare than the Turnover syndrome, but little is known about it. The frequency is approximately 1 in 1,000 female infants that occurs where the commonly when the mother is ordered. Okay, these are the Turner syndrome, triplex syndromes. The characteristics of the triplex syndrome: the web skin, next is the construction of poor breast development, especially underdevelopment ovaries. Okay, next one is the Klinefelter syndrome, is also male sex chromosome abnormalities. Males inherit one or more extra X chromosomes. Their genotype is the XXY, or rarely triple XY, or XY oblique XXY mosaic. Okay, in different, especially X chromosome is the double in the Klinefelter syndrome. In services case, they have the relatively high pitched voice. Asexual is the feminine body contours, as well as the breast enlargement and the comparatively little facial and the body hair. These are the sterile are nearly so, and there is the testes of the prostate gland are small. Next one, the produce of the relative small amounts of the testosterone. They also like to be the overweight. They usually have the learning difficulties as children, especially with the language and short-term memory. They unable to produce sufficient amounts of the sperm for the conception. This may be connected to the low testosterone production, especially these are the mainly some the characters related to the women characters. This may be the okay. These are the Turner syndrome, Klinefelter syndrome. Main is the X chromosome, allosomal, especially sex chromosomes, male sex chromosome deficiency. Next one, low testosterone, especially the frequency of the Klinefelter syndrome has been the reported to be the between 1 in 500 and the 1 in 1000 male birth. This makes one of the most common chromosomal abnormalities. The males with the Down syndrome sometimes also have the Klinefelter syndrome. The both syndromes are the more likely occur in the babies in the older mothers. As a result of the produce relatively small amounts of the testosterone, this the feminizing effects of this the hormonal imbalance can be the significantly dismissed to Klinefelter syndrome. Boys are the regularly given testosterone from the age of the puberty on. Okay, the frontal, these are the Klinefelter syndrome characters. Some of the male characters, the poor, especially breast growth also is very poor. Tendency of the grow is a fever chest hair. Next one is the narrow shoulders, and next is the small testicular size, long legs, and next is the female type of the pubic hair in the pattern. Okay, XY syndrome, XYY. If males inherited extra Y chromosome, there is the genotype XYY. The adults, these are the super males, are the usually tall, above the 6 feet, and generally appear to the normal. However, the produce high levels of the testosterone during the adolescency. They often are the slender, have the several facial icon, and the poorly coordinated. XXY chromosomes, males are the male. Already we types of the chromosomes is over: telocentric, acrocentric, metacentric. Metacentric also centromere is the middle. Submetacentric is the centromere of century leading the short pure relatively. Acrocentric, acrocentric is the eye shape. Next one, telocentric is the these are the telocentric. These are the diagrams of the telocentric, acrocentric, and the submetacentric, mainly based on the position of the centromere. Based on the position of the centromere. The morphology, chromatid, the chromonymata, the chromomeres. What are the chromomeres? These are the deep strained bed-like structure present along with the length of the chromosomes, and are the visible of the interface of the chromosomes, and also packed in chromosomes of the meiosis. The structure that is the aligned side of the slide in the polyethylene chromosomes, the resulting of the banded appearance in chromomeres. It is the thought they are the arise due to the difference in the coiling of the chromosomal filaments at the earlier stages of the cell division, while they are the longer visible in the metaphase chromosomes due to the tight coiling. Already we discussed the centromere. Centromere also different types of the centromere. Is the mainly is often referred to the to as the kinetochore. It is the reason that the chromosomes remain attached to the mitotic spindle. The centromere lies within the thinner segment of the chromosomes. It is called the primary construction. It is known known that the regions flanking of the centromere frequently contain high, highly repetitive DNA, maybe strain more the intensely due to their the heterochromatic nature. Telomere, already we discussed the telomere. Next one is the telomere, especially the term applies to the tips of the chromosome. Telomere is the term apply the tips of the chromosome. It is the region that the prevent the chromosomes being circularized. Centromeres also to the broken radiation treatment only on the sticky ends can fuse, but the fusion of their non-sticky kilometric ends and not preserved. The chromosomes send in the in all eukaryotes have the specific short DNA sequences present as well as a small cluster of the several repeated units are the duplicated only through the special enzyme is called as the telomerases. Special enzyme is also called as telomerases. Next one, nuclear organisms, the secondary construction of the chromosomes. Secondary construction, in addition of the primary construction, sudden the chromosomes bear the secondary construction. The structure, if the present is the usually seen in the distal region and the arm, the small fragment being the pitched off from the cord is the turbulent and satellite. The satellite remains attached to the rest of the body thread chromatin secondary. Secondary construction. So next one is the nuclear organizer. There the secondary construction region and the some chromosomes are is also called as the nuclear organizer regions, and the chromosomes carry them are called as the nuclear organizer. They contain the genes coding for the 18S and 20S ribosomal rRNA. Ribosomal RNA. It can induce the formation of the nuclei. The secondary construction mainly the secondary construction region may be arise because the rRNA and genes that transcribed very actively infecting the chromosomal condension. The chromosome bearing the regions are the specific in each organism, as the seventh chromosome complement in the parallel millet of the nuclear organism chromosome. The bearing satellite regions are the described mainly satellite chromosome. The satellite is the rounded body separated from the rest of the chromosomes by the secondary construction. Next one is the karyotype. What is the meaning of the karyotype? Karyotype means the set of the features like chromosomes, length, centromere position, and the other reproducible components of the chromosome that allows the identification of the chromosomal complement. The species is also called as the karyotype, especially the set of the chromosomes. Mainly, these are the set of the chromosomes is also called as karyotype. Set of the chromosome. Some of the more advanced eccentric karyotype represent a large difference between the largest, smallest chromosomes, variation centromere. What are the chemical composition of the chromosomes? Mainly, these are the chemical composition of the chromosomes also, different types of the chemical composition. What are the main chemical composition of the chromosomes also? The chromosome DNA, RNA is the main. Mainly chemical composition of the chromosomes are the DNA and RNA and some of the proteins also main chemical composition of the proteins. The proteins present in the chromosomes could be majorly classified in the histone and the non-histone proteins. DNA, main DNA is the most important chemical component of the chromatin. Is are the quantitative measurements of the DNA have been made of the large number of organisms during the late 1960s and is the related the recent genes in 1972. The quantity as always expected the picograms and is the and the mainly the 31 centimeters of the double helical DNA. It has the found the quantity of the DNA varies greatly different organisms. The DNA content is the applied complement. The species referred to it's the genomic DNA. It is the smaller in the bacteriophage. Actually 750 kilobyte bases also in bacteriophages. The small weight of the bacterial chromosomes also they can see into the in bacteriophages. The bacteriophage is also the virus. So the DNA content is haploid complement of the species is referred to the genomic DNA. It is a smaller DNA. Next one, the bacterial DNA is the measures from the 1.1 to the 1.4 mm. And next one is the histone proteins. The histone proteins is the kind of the basic proteins which have the 5 subfractions H1, H2A, H2B, H3, H4. These are the 5 types of the histone protein. And H3 and H4 is the highly conserved, having the same structure in different species. These are the common functions H3 and H5. Next one is the histones of all these types of the shown considerably similarity in different organisms with the much conservation during the evolution purpose. The features is that the proteins should be play the structural role rather than the regularity of the although more recent studies attributed the these are the proteins. Non-histone proteins also there's the non these are the non-basic proteins present in a limited extent in the chromatin, although variety of them could be traced to the different organisms. Different organisms. Heterogeneity of these proteins suggested the conserved the evolution stones. The non-histone proteins differ even the between the different tissues, organisms. Model of the DNA folding, different types of the models of the DNA folding. Mainly is the study under the electron microscope, structural study of the chromosomes, important in 1960s. Mainly, the DNA is the 2 nanometer wide, and the possible the fibril may correspond to the single DNA molecule. If the single chromatid has the single long DNA molecule has been provided later, nucleosome model. Nucleosome model also mainly these are the seen under the microscope and electron microscope. Mainly electron microscope studies the chromatin from interface nuclealized in water have shown the chromatin to be fibrous with the arrays of the spherical particles. Electrophotometric and the gel filtration techniques have the relieved the extension of the 5 stone protein. Already we discussed the H1, H2A, H2B, H3, H4. Already is the H3, H4 is the highly conserved uh, histone protein. These are the chromatins are eukaryote involved in the organization of the nucleosomes. Nucleosome. Some with the chemical cross-linking histone proteins was found in the H3, H4 are present in the tetramer, while the H2A, H2B is associated into the another treatment. These are the next one is the solenoid model, nucleosome model. So different types of the models. Solenoid model it was the later shown 11 nm fiber is the nucleosome gets the coiled upon the itself from the 13 nanometer, where the helix is the 5 or to 6 nucleosomes for the helix. In this helix, successive turns become the close on another. That is the center distance about the 10 nanometer. This 30 nanometer structure also called as the solenoid. The term in physics, solenoid means the coil on a central axis. Coil as a central axis. Solenoid means coil as a central axis. Your book in also 53 page 3.2 diagram is the solenoid model. Next one, loops and domain. Scaffold the chromatin. The scaffolding means folding nature. Next one is the euchromatin and heterochromatin. What is the mean of the heterochromatin? Is there the routine fixing stain procedure observable that the different parts of the chromosome undergo the different condension cycles. It is the interface is the certain highly condensed some of the which is also called as the chromocentromeres. H in the ninth kind, the term heterochromatin then is the chromosome regions which is the remain the highly condensed state. These are the less strained regions as the euchromatic ones. Reasons showing the heterochromatin properties have been the broadly categorized are the two types: constructive heterochromatin and the facultative heterochromatin. Constructive heterochromatin it is usually present in the same position of the two homologous chromosomes and inherited within the chromosomes. It has been the found to the occur at specific regions and are termed accordingly. They are the centromeric near to the centromere. The facultative heterochromatin is often seen the develop during the development an organism. An example when the entire chromosomes becomes the heterochromatic provided by one of the X chromosomes is the mammalian female. One of, one of the two X chromosomes in the female remain euchromatic throughout the life cycle, while the other X in the female becomes heterochromatic in the course of development from zygotic to the embryo and takes up the distinctive staining. What are the functions of the heterochromatin? Mainly, they are the heterochroma finally lead to the different functions organ. The heterochromatin may be lead the some organisms by the nature and accident of the heterochromatic segment is translocated to next to euchromatic uh, structure. There are the heterochromatin and euchromatin. Next one is the special type of the chromosomes. What are the special types of chromosomes? The chromosomes are highly variable in some tissues like the salivary glands of Drosophila, oocytes of the amphibians, salivary glands of the Drosophila, and next one oocytes of the amphibians in some of the invertebrates. The special category called beta chromosomes were also reported in the many plant species that the devoted to perform different functions. The salivary glands of the chromosomes. See, this one is the special type of the chromosome. What are the special type of the chromosomes? And in eukaryotic organisms, certain the chromosomes are the found only in certain special tissues and are not the seen in the other tissues. Especially they are the available seen to the special type of the tissues. These chromosomes are larger in size and the call is the giant chromosomes. In certain plants, they are found in the suspension of the embryo. There are two types of the giant chromosomes. One is the Paltin chromosome, another one is the lamb brush chromosome. What is the main giant chromosomes? The chromosomes are the larger in size, and are also called as giant chromosomes. Okay, next one is the Paltin chromosomes. Paltin chromosomes are absorbed by the C.J. Balbani in 1881 in the salivary glands of the Drosophila. And Drosophila melanogaster is the flying flying mosquito. Okay, especially these are the Paltin chromosomes observed the C.G. Balbani in 1981 in the salivary glands of the Drosophila. What are the characteristic features of the Paltin chromosomes? These are the along the length of the chromosomes. There is a series of dark bands alternate with the clear zones called interbands. These are the Balbani chromosome large. Okay, these are the Paltin chromosomes. See, this one is the diagram also Paltin chromosomes. Paltin chromosomes are the extremely larger of called is the Balbani ring. These are the Balbani ring. See, the Balbani ring also large size of the Balbani ring. It is also known as the chromosomal puff. As this chromosome occurs in the salivary gland, it is known as the salivary gland chromosomes. What are the uses of the Paltin chromosomes? Paltin chromosomes. Mainly these are the seen the salivary glands of the chromosomes. Drosophila in Drosophila. The glands about the 1,000 DNA molecule arranged side by the side, a result from the rounds of the DNA replication. In each strand or fibril, there are the some regions is called chromomeres, in which is the chromatin is more tightly coiled with the alternate with regions where DNA fibers are the folded more loosely. Thus, would be the series of the dark bands alternating the clear zones is called interband. Interband. There are the many as 5,000 bands of the Drosophila genome. The details there, the morphology and the position of the chromosomes feature of the identification. And the material offers advantage in the cytogeneticity such as the chromosomes mapping. Paltin chromosomes I have the provide the first evidence of the gene activity regulated at the level of RNA synthesis. Then the available material is but the mainly is at the level of RNA synthesis. RNA synthesis mainly is the valuable material for the study of the gene regulation because of the transcription of the gene can be visualized directly in the microscope. These specific sites on the chromosomes with the genetic activity is called puffs. The puff can be considered as a band in which is the DNA unfolds into the open loops as the consequence intensive gene transcription. Berman 1952 has studied extensively puffing different tissues of the cynos under the natural and the treatment of condition. Puffing cyclic tissues developed the specific reversible phenomena. Paltin chromosomes are very suitable in the in situ DNA hybridization because the thousand of DNA molecules are the aligned side by the side, greatly helps in the detection of the single gene copies. What are the importance of the uses of the Paltin chromosomes? Mainly, Paltin chromosomes are usually found in the larvae, whereas the believed these many replicated chromosomes allow for the much faster larval growth than if the cells remain the diploid. The Drosophila Genome Project uses the 14 chromosomes as the framework for the mapping. Mainly the Drosophila Gene Project also uses the 14 chromosomes the framework of the mapping. Next one, lamp brush chromosomes. These are the special kind of the chromosomes found in the primary oocytes of the amphibians. They are the very important. It is the spermatocytes nuclei insects, including the Drosophila, and also in the giant nucleus of the unicellular alga, Stabularia. That the first observation of the Fleming and later is shark. Mainly the first observed the Fleming in 1982. Later shark oocytes the Rocket 1992. 92. These chromosomes are seen during the prolonged diploiding stage of the metatic prophase in these cells and may have the sometimes attain up to the length of the 1 mm to as the urinal amphibians. Mainly the Lamprey chromosome is a special type of the chromosomes. Mainly these are the seen to the oocytes of the amphibians. Oocytes of the amphibians. Lamprey's chromosomes are the first observed by the Fleming in 1882. Is looked from the brush-like structures. They occur at the diploiding stage of the mitotic and prophase oocytes of the animal salamander, and it is the giant nucleus of the nuclear alga Stabularia. The highly condensed chromosomes forms the chromosomal axis from which is the lateral loops of the DNA extend as the results of the intensive RNA synthesis. Mainly the main important is the RNA synthesis. Already special type of the chromosomes also are polytin also. Polytin chromosomes also the gene activity regulated the level of the RNA synthesis. Okay, RNA synthesis. Okay, the chromosomes show the chromatophore pattern, which is the pairs of the loops projecting out from the majority of the chromosomes. Since these chromosomes are the seen in the metatic prophase, they are the present in the form of bivalence with the material and the pattern homologous being held together the chiasma of the regions. The crossing over is the occurred in previous mitotic regions. The chromosomes also represent the row of granules of the axis, each homologue from the which lateral loops extend. The loops are always symmetrical with the pair of the loops in each chromosome, also with the additional symmetrical pair on the other. Uh, it it is these loops are the give the characteristic appearance of the chromosomes, such as the rocket named in the Lambrecht chromosomes. Lambrous chromes like these are the like the brush used in the cleaning of the chimneys of the oil lamp. Okay, Lambrous chromosomes. Lambrous means the brush like structure. The size of the loop varies within the average 10 micrometer with the 9 to 9 sorry 1 to 9 such loops are the for each chromosome. Each loop appears to the constant position in the chromosomes, and there can be many as 10,000 loops each for each set of the chromosomes. The size and thickness are the morphological characters of the appear of the identifying features. That is the detailed chromosome maps in the drawn from the other one. And the loops are the sites active genes and the contain RNA. Each loop has an axis formed by the single DNA molecule that is the unfold from the chromosome during the DNA synthesis. Each loop has each loop is covered by matrix that contains RNA and transcripts with the proteins attached in them. In general, the ribonucleoprotein matrix is the asymmetrical, being the thicker at one end of the loop than at the other end. Some structure of the typical Christmas tree pattern with the nascent ribonucleoproteins chain attached to the perpendicular DNA axis. Many loops correspond to the single transcription unit to show the continuous in the like salivary glands chromosomes, that is the Lamprecht chromosomes. Also, a very good material of the studies of the institute DNA hybridization because of the thousands of the nascent RNA molecules aligned side by side along the loop amplify the hybridization signal. Next one is the beta chromosomes. These are also called as the supernumerary chromosomes. Beta chromosomes are accessory chromosomes. These are also called as the supernumerary chromosomes. They can be used to define extra chromosomes that are not necessary for the individual living. They are not homologous to any part of the normal chromosomes. These chromosomes differ from the normal ones. There is the number, smaller size, and the degree of the heterochromatin. They may vary in number within the different tissues of the same individual between different individuals of the species in population and even between the different population of the species present in the different localities. They are usually acrotolocentric in nature, although few are the metacentric. They do not usually pair of the normal chromosomes during the meiosis, but the pairing of the and so show the formation of the chiasma. The first recorded maize in the Longley and the Rodolphish 1927 to 28. Addition of the 2n equal to 20 chromosomes the complement. They are the much smaller than the A chromosomes, normal chromosomes, and the varied from the 1 to 20 nanometers. They are the tightly heterochromatic surface and having the differential staining cycle. This kind of the chromosomes have been reported in the subsequently many plant systems, spite of the system. In most of the case of the de deleterize, when the present of the higher number and the influence of the morphology, the plant as well as the chromosome behavior during the meiosis, the also effect of the A chromosome pairing, chiasma formation and crossing over. The most significant of the beta chromosomes on a pollen fertility, when is are seen to the pollen fertility and seed set, the the flowering generally delayed and ray plant and further decrease the chiasma formation and the presence of the higher numbers. A number of A and B translocation have been the reported in the and maize, which is the indicate the influence of recommendation frequency of the A genes and indicates the different segments of the beta chromosomes influence differently the crossover presentation. Much of the work homologous pairing of the A chromosomes and the influence by the BS in the wheat into Agelopsis, wheat ray and hybrids. The beta chromosome appear in the positive role in the and main important is the positive role in the plant breeding techniques. What are the importance of the use of the Lamprecht chromosome? This is the formed due to the active synthesis of mRNA molecules for future and use of these egg cells when no synthesis of RNA molecules is possible during the mitotic cell division. This is the special type of the chromosomes, beta chromosomes, and next one is the alpha chromosome, giant chromosomes, Paltin chromosomes, Lamprecht chromosomes, the different types of the chromosomes. Okay, these are the types of the chromosome. And this classes will mainly discuss chromosome types. What are the chromosomes, types of the chromosomes, next one chromatins, uterochromatin, euchromatin. Mainly heterochromatin is the area of the chromosome which is the darkly stained with the DNA specific stain and is the comparatively condensed from the condensed form. Heterochromatin main, what is the main important of what is the definition of the heterochromatin? Is as the area of the chromosome which is the darkly stained with a DNA specific stain and is the comparatively condensed form. Next one is the euchromatin is defined the area of a chromosomes which is the rich in genes that they actively participate in the transcription process. Mainly, mainly heterochromatin the mainly darkly stained with the DNA specific stain and is the comparatively condensed form. Euchromatin defined as the area of chromosomes which are the rich in a genes that they actively participate in the transcription process. Okay. There the total theory and summary of types of the chromosome, chromosomes types, chromosome definition, heterochromatin, euchromatin, the special type of the chromosome, Partin chromosome, giant chromosomes, beta chromosomes. These are the chromosomes. Thank you. Okay, next topic is the cell cycle and apoptosis. Uh, Cell cycle and apoptosis. Next one is the what is the mean of the cell cycle and the apoptosis? Um, all somatic cells, the multicellular organization, are the descendants one of the original cell, the fertile eggs, the zygote through the cell division. An adult human is the as being the approximately 1 lakh billion cells. All organisms form the single cell, the fertilized egg cell. Somatic dividing cells undergo the process termed as the cell cycle. Somatic dividing cells undergo a process termed as a cell cycle. The cell cycle is an ordered series of events leading to the cell replication. The goal of the cell cycle is to the produce two daughter cells that are the accurate copies of the parent. In multicellular organisms, most differentiate cells exit cell of the cell cycle and survive for days, in weeks, in some cases. Such cells are exit and the cell cycle. Cell cycle research has primarily been performed on two strains of the yeast, the budding yeast Saccharomyces cerevisiae and the fusion of yeast Saccharomyces form. Cell cycle and apoptosis. 1 minute. Okay, cell cycle and apoptosis. ready to the prepare for the M phase. G1 phase is over, S phase and G2 phase also is over. Next, then I'll go to the N phase. M phase is the karyokinesis included prophase, chromatid coiling, disintegration of nuclear in prophase 1, the membrane and the nucleus spindle formation, metaphase, chromosomal orientation at the equatorial plane, anaphase, movements of the chromatid towards the opposite poles. They can inform the metaplate, telophase, that the reconstruction of the daughter nuclei. The reconstruction of the, in especially M phase also they are containing the two subcategories, karyokinesis and cytokinesis. The karyokinesis is the prophase, metaphase, anaphase, and telophase. And the cytokinesis including the formation of cell plate, the leading the equal division of the cytoplasm, nuclei, cell organelles, and cell membranes into the two daughter cells. See the diagram is the formation of the cell plate. After cell division, each daughter cell begins the interphase in new cycle. Some cells are the heart, kidney, liver, neurons. After remaining the G1 phase for the some time, come out of the cycle. Now the okay, after cell division, the some cells of the cells in a quiescent phase of the cell division stops the activities of the cell continue. Sometimes the cell re-enters the cell cycle from the quiescent phase when required. For example, during the formation of the periderm, what are what are the regulation of the M phase? Next one, regulation of the cell cycle. Regulation of the cell cycle. Cell cycle does not occur to the unchecked the presence of the cells checked by the regular molecules. It includes the detection and repair of the genetic damage as well as the prevention of uncontrolled cell division. There are the two key classes regulate the molecules of the determined cells proper progress through the cell cycle. These are cyclins, these are the proteins. Next one, cyclin dependent kinase, CDK. See, this one is the cyclin proteins. The diagram also cyclin protein and CDK. Cyclin protein 1, 2, 3. And the CDK also CDK. Mainly cyclin proteins also the space factors of, and the space begins in the DNA amplification enzyme attach. What are the phase of the cell cycle? The G1, S, G2. What are the cyclin proteins? Cyclin D. Also, they can see into the in G1 phase, in G2 phase. cyclin E and cyclin A, and G2 phase cyclin B. CDK also present in the checkpoints in CDK G1 phase CDK4, in S phase CDK2, in G2 phase CDK1, in cyclin CDK complex in G1 phase, G1 cyclic and G1 CDK, and next one S phase, cyclin S phase CDK, and G2 is the mitotic cyclins in M phase CDK. What are the functions of the CDK and cyclin proteins? Mainly in G1 phase, inhibits the RBA protein and signals the cells of the prepare the chromosome for the replication. Activities of the DNA replication in S phase, especially in replication, is the main important in the DNA synthesis in S phase. What are in G2 phase? Activities the mitosis. What are the cell cycle checkpoints? Cell cycle checkpoints are used by the cell monitor and regulate the progress of the cell cycle. The cell cannot proceed to the next phase until checkpoint requirements have been met. Two main checkpoints are the G1S, G1-5 checkpoint before all enter into the S phase, G2/M checkpoint after S phase, G1/S checkpoint is the before enter into the S phase, checks for the cell size, checks checks for the nutrients, checks for the DNA damage, checks for the all the preparation, all proteins, the requirements of the S phase. Next one, checks whether S phase has the cyclin CDK complex. CDK complex. Okay, next one, G2 checkpoint after S phase, before S phase, after S phase. Checkpoints prepare the DNA replication. Checks for the all preparation of the all proteins ATP required for the M phase. Checks for the tubulin synthesis. Checks whether the M phase cyclins and the CDK complex related to the initiative mitosis. Then the cell passes to the next M phase, cell death. Cell death is an important process in the body as the promotes the removal of the unwanted cells. Cell death is also called as the apoptosis. The cell death is the event biologically cell stop to carry out of the functions. This may be the results of the natural process of the old cells dying and the being replaced by the new one. Cell death can occur in following ways: apoptosis, cell suicide, or autophagy is also called cell suicide. Necrosis is the cell murder is one type of the apoptosis is the autophagy are both forms of the programmed cell death. while the necrosis is the physiological process of the occurs the results of the infection or injury. Apoptosis is the if cells are the no longer needed, they committed suicide by activating and the intracellular death program. This process is the therefore called programmed cell death, although it is the more commonly called apoptosis. For an average human child between the ages 8 to 14 years old, approximately 20 to 32 billion cells are die for the per day. Apoptosis is a highly regulated process. Apoptosis can be initiated through the one of two pathways. In the intrinsic pathway, cell kills itself because it's the senses cell the stress, while the extrinsic pathways, the cell kills the itself because of the signal from other cells. In regulation, these are there are the variety of the factors responsible for regulating apoptosis, intracellular and extracellular. Intracellular means DNA damage or failure of cell division. Intracellular, extracellular growth factors are specific signals from the other cells. Growth factors, specific signals from the other cells. Apoptosis inducers, withdrawal of the growth factors, some viruses, free radicals, ionizing radiation, DNA damage. These are all are the inducers. Ligand binding death receptors. What are the apoptosis? Which are the two pathways, intrinsic pathway and extrinsic pathway, apoptosis pathways. Intrinsic pathway, activation of apoptosis via mitochondria in intrinsic pathway, where stress signals and DNA damage signals and defects of the signaling pathways are processed. The intrinsic pathway uses the mitochondria as the central component for the activation of apoptosis. The diagram is the intrinsic pathway. Okay, next extrinsic pathway uses the extracellular death ligands, Phage ligand, tumor necrosis, the factor TNF death receptors, which pass the apoptotic signal, the integrated campus, and it is the exogenic campus. In the excretion phase of the apoptosis, various cellular substrates are degraded, leading to the cellular collapse. As a consequence of the caspase activation, a number of key enzymes and structural proteins of the cell are the degraded, leading to the cell death. The stimuli that the induced apoptosis are very diverse and they include, for the example, DNA damage, stress condition, malfunction of the pathways regulating the cell proliferation. Necrosis also, cell death is one type of the some total of the morphological changes of the follow cells death in the living tissue of an organism. Dead cells usually show the changes of the both in the cytoplasm and it is the nucleus. Cytoplasmic changes are the increase the eosinophilia, glossy appearance, granular voice increase, different types of the mechanism of the necrosis are involved, loss of the metabolic functions, loss of integrity of the cell membrane, cassation of the production of the proteins and ATPs, cell organelles swell and they become the non-functional. Necrosis for autophagy also. What is the meaning of the autophagy? Autophagy is the self-degradative process that is the important for the balancing success of energy at critical times in development and the response to the nutrients stress. The liver is a rich lysosomes and the process possesses high levels of the metabolic stress induce the autophagy. Autophagy regulated by the correction of the hormones and the amino acids. These are the cell death and apoptosis, cell cycle, different mechanism of the cell cycle. Checkpoints also very important. What are the reason for the programmed cell death? Is mainly cells having the no function, death harmful cells, development of the defective cells, elimination of the excess cells, cells that have the finished their the function. These are the main reasons of the programmed cell death. Changes mainly genes controlling the apoptosis, some of the um, nematodes. In this process, some of the following stages are followed. Dissection, whether the cell has to the survive the or die. Execution of the cell death. Engulfment of the dead cells in the phagocytosis, degradation of the cell. Mechanism of the apoptosis and the cell regulation apoptosis, carbohydrate signal apoptosis, signal, meals. All are the mechanisms for the apoptosis. Thank you.